Welcome to part 2 of the Protozoa lecture. In this lecture, I'll go over the life cycles of Giardia lamblia and that of Plasmodium. So let's start with Giardia. Giardia belongs to a group of flagellates that lack a mitochondria. Now remember that mitochondria is very important in our bodies and our cells because it serves as the powerhouse of eukaryotic cells. That's where our ATP is made during cellular respiration. So instead of having mitochondria, these organisms have kinetoplasts, which are modified mitochondria. Thus, these protozoa are called kinetoplastida. This is just another example of a unique feature that is found among protozoas and not in other eukaryotic organisms. In the US, Giardia is the most common human intestinal parasite. It is transmitted through contaminated water, and it causes the diarrheal illness called giardiasis, and it's often transmitted by a fecal to oral route. Giardia can be found in the stools of infected humans and animals, mostly in animals, such as dogs, cats, beavers, and rodents. In this image, it's found in your textbook on page 197. It shows hundreds of trophozoites that are attached to the insides of a gerbil's intestines, which are actively feeding, and most likely they will be dividing by fission. So remember that fission is an asexual form of reproduction. It lives symbiotically in a commensal manner in the small intestines of humans and other mammals, but sometimes it can become parasitic. It's often excreted through the feces as a cyst or a trophozoite and survives in the environment as a cyst only before being ingested by the next host. Now, people who drink untreated surface water from ponds, lakes, streams, and rivers have a higher risk of acquiring giardiasis. This mode of infection occurs mostly among backpackers, campers, or others who are in the habit of drinking untreated stream water. So you can imagine you'd have a wild animal who would have giardia in their intestines. They would defecate in the water, therefore there would be some cysts present in the water. And if a backpacker or a camper didn't treat the water through filtration or boiling before drinking, that's how they could acquire the protozoa inside their intestines. However, this rate of incidence is very low. Another way that we can acquire giardia in our intestines is by eating or drinking food or water that's contaminated with human or animal stools that are contaminated with the parasite or its cysts. It says eggs in the slide, but it should be cysts. This is a figure from the CDC webpage, and it basically shows, again, contamination of water, food, or hands or fomites. Fomites are inanimate objects that would be infected with cysts. And they're showing you a person ingesting the cysts. Okay. So once the cyst enters, it has to enter the stomach, and then it lodges itself into the small intestines. That environment switches the cyst into form a trophozoite. The trophozoites then multiply, as I mentioned, by fission. They survive there for multiple rounds of asexual reproduction. A signal turns them into cysts, and they are released from the body, and then the whole life cycle continues. This is also a risk factor for people who work in daycares and nursing homes. So you can imagine changing diapers and not washing hands in between infants. You can then spread the disease or the parasite from one person to another, including yourself. So this slide is just to remind me to tell you that the highest rate or the highest chance of you acquiring giardia is often by transmission in a daycare setting. So daycare centers from preschool age children and institutions for the developmentally disabled, this is where most of giardiasis does occur in the US. What are the symptoms? The symptoms actually include frequent loose, pale, or greasy stools, often foul smelling because of the metabolic processes or waste that the protozoas create. You may feel some bloating, some cramps, fatigue, and weight loss. Fever is usually not present, and many people infected with giardiasis, or giardia I should say, might not even have any of these symptoms at all. So they might be just commensal, meaning that the giardia benefits from the host, but the host doesn't benefit at all from the relationship, or it might be a little bit more parasitic in nature, in which these symptoms do occur. Okay, you should be familiar with this slide. I showed it to you in part 1 of this lecture series, where I introduced the ciliates, the amoebozoans, and the flagellates, with giardia being one of the flagellates. However, there's a fourth group of protozoas which aren't represented in this figure, and they're called the epicomplexans. Now, members of this group of the epicomplexans have a more complex life cycle with several stages and often more than one host. They reproduce both sexually and asexually, but their asexual form is not fission, but in this case it's schizogony, which I've mentioned in part 1 of this lecture. They often rely on vectors to transmit them from one host to another, which makes their life cycle a lot more complicated than of that of giardia as an example. They all have a unique physiological structure at the apex end of their cells called the apical complex, hence their name epicomplexans. This apical complex allows the organisms to enter and replicate inside a host cell, and they're very specific which host cell that they can enter. So for example, when I talk about Plasmodium, the protozoan that causes malaria, this apicomplex will allow the Plasmodium to enter either red blood cells or liver cells. So as a result, these protozoans will often cause damage to cells, and they will be considered parasitic in nature to their hosts. An example of the apicomplexan protozoan is that of the Toxoplasmosis gondii, which causes the disease toxoplasmosis. Now, this parasite is mostly found in domestic cats. They're the only known definitive host where the parasite undergoes sexual reproduction. But it's easily transmitted to humans or other warm-blooded animals where we carry it in our body as an intermediate host. I'll give you more of a definition of an intermediate and definitive host when I talk about Plasmodium. Which gets me to the other example, and that is of the Plasmodium protozoan. Now, Plasmodium is the genus name, and there are 4 species of Plasmodium that's associated with causing malaria. Now, your textbook refers to a study that recorded 584,000 deaths worldwide in 2013. Now, they didn't break that down any further, but I've also found a reference of a study in 2015 that recorded 212 million total number of cases worldwide. Of those cases, there were 429 deaths worldwide, with 92% of those cases only centered around in Africa. And of those cases, 303,000 deaths were associated with children under the ages of 5 years old. So it gives you an idea that there are certain regions of the world that are more prone to malaria, whereas others it would be more of a rarity. So between 2010 and 2015, the malaria incidence rates fell by 21% globally, and in that of the African region as well. During the same period, malaria mortality rates fell at an estimated 29% globally, and by 31% in the African region. So you get the idea that most people are focusing about the African region because that seems to be where predominant malaria occurs. Now, I'd mentioned that Plasmodium is the genus and that there are 4 species that are associated with malaria, and I've listed them here. There's Plasmodium falciparum, Plasmodium malaria, Plasmodium ovale, as well as Plasmodium vivax. Some of these are more virulent or more pathogenic than others. Now, the Plasmodium life cycle is going to be very complex. It's going to have 2 types of trophozoites. They are shown here in this slide as the form of a sporozoite and as a merozoite. Now keep in mind that the sporozoite is mostly associated with the mosquito. It's what is injected inside the human, but then once in the human it is converted into a merozoite. So let's talk more about these terms. The sporozoite is the infectious stage. It's associated or found in the mosquito, and it's the form of the protozoa that's transferred to humans. Again, once it comes into the humans, it's going to divide asexually. It's going to do this by schizogony, and the resulting progeny, the daughter cells, will be called merozoites. These merozoites, just like the sporozoites, are going to be trophozoites because they will actively grow and divide asexually. There's going to be a ring stage. The ring stage are actually the merozoites that are inside the red blood cells. Now, if you look at them, they'll have a ring with like a little round circle at the end. It will actually look like a stone at the end of a ring. So histological stains, they look like there's a ring inside of a red blood cell. So we call this the ring stage. You also need to know about the two hosts. There's the definitive host and the intermediate host. The definition of a definitive host is the host in which the protozoa reproduces sexually. In this case, it will be the mosquito. The definition of an intermediate host, again, is the host in which the protozoa only can divide asexually. For that of Plasmodium, it's the human. As well, it could be other warm-blooded animals also. As mentioned, the form of asexual reproduction is going to be that of schizogony. You might need to go back to part 1 if you've forgotten what the term schizogony refers to. And the definitive host is the mosquito. It's going to be the Anopheles mosquito, which we also sometimes call as the vector. Okay, so let's talk about the life cycle of Plasmodium. I'm going to start with it as a sporozoite inside the mosquito, inside the salivary gland, and here comes a mosquito that is biting a non-protected human. In this case, the gametocytes, they are the male or female form of the haploid cells, like the egg and the sperm in humans. They've already fused together in the gut of the mosquito and have migrated to the salivary glands of the mosquito and are injected as a form of a sporozoite inside the blood cell during a blood meal. So I have here that the infected mosquito bites a human. It injects the Plasmodium sporozoites inside the bloodstream. And that bloodstream goes to the liver, which the liver acts as a filter of the blood. So the sporozoites, when they reach the liver, they have the apicomplexa that allows them to enter inside the hepatic cells, or the liver cells, where they can undergo their first round of multiplication by schizogony. The resulting daughter cells, as shown here, here's a sporozoite entering a hepatic cell. It's undergoing a round of schizogony. Eventually, the whole hepatic cell is going to burst open because it's going to make hundreds of merozoites. Remember, merozoites are the progeny. Now, I said hundreds. I don't actually know what the number is, but as you can tell by the picture, there are many. As the liver cell ruptures, these merozoites are able to re-enter the bloodstream. Now, they're no longer sporozoites, so the apicomplexa allows them not to enter back inside the hepatic cells, but allows them to enter into the red blood cells. So these merozoites are going to be in the bloodstream. They're actively looking for red blood cells in which they can enter. So RBC stands for red blood cells. So here's an oversized artery or a vein. These are red blood cells, and you're going to see the merozoites enter inside a red blood cell right here. As the merozoites are dividing, at one point they're going to look like a ring. So here's a picture of a ring stage, and here is a histology stain. I'm showing you it looks like the band of a ring with a jewel or a gem right here. So this is a good example of a ring stage. At this point, these merozoites are going to continue to divide by schizogony, and they're going to be released. They're called schizonts here, but they're actually going to give off to more progeny, more merozoites. As the cell ruptures, these merozoites will continue to multiply. They'll enter other red blood cells. As you can see, that this is a vicious cycle that can continue to many, many rounds of replication. Okay, this is a very nice picture. It's an electron micrograph. These are red blood cells that are healthy, and here are infected red blood cells and showing you a merozoite. Okay, so we're back at this slide here, and here's the merozoites. As these merozoites are dividing, they're also releasing some waste product in the blood, which will add to symptoms that the patient will feel. One of the symptoms is that of chills and fever due to these waste products. Now, what effect does Plasmodium have on the humans besides chills? They'll actually feel acute febrile illness, so they'll be a little weak. The symptoms will appear 7 days or more. Usually will last a little longer, 10 to 15 days after the infected mosquito bite. The first symptoms will be that of fever, headache, chills, and vomiting. May be mild or difficult to recognize as malaria because it sounds like symptoms of other diseases also. If not treated within 24 hours, though, it can progress into a more severe illness, and it can also lead to death, as given by the statistics that I showed you earlier. So it is considered as a life-threatening disease. Now, 2 types of merozoites will follow. Some will infect new red blood cells, as I showed you here, continuing this circle of continuing infection. Others will develop into a male or female gametocyte. So here's my gametocytes. One is a male and the other one's female. These will stay in the red blood cells, and as long as they stay in the red blood cells, nothing will happen. Now, these are going to be haploid in nature, and they need to fuse together to form to continue or to finish the sexual form of replication. So we're showing you here up top the male and the female gametocytes. Once they enter the gut of the mosquito, they fuse, so they go from haploid to diploid, and they form a sporozoite. But this stage here, this fusion stage, does not happen in the human red blood cells or the human blood. It only happens when a new mosquito comes along, and during a blood meal will ingest the gametocytes. Otherwise, the gametocytes are a dead end inside the human. That's why the protozoa only replicates asexually inside the human and can replicate sexually inside the mosquito. Here's just another picture of Plasmodium falciparum. It's the most deadliest of the 4 species. It's also the target of vaccines. Plasmodium vivax is secondly to its parasitic in nature, but is not part of the vaccine yet. Again, here are normal red blood cells. Here's an abnormal red blood cell that is clearly is infected with something. You will know histologically if you have Plasmodium in your blood by doing a blood smear, and this is what a blood smear would look like. These are obviously ring stages, and these are the sporozoites that entered that have not become have not really reached the liver yet and turned into merozoites yet. Okay, other common parasitic protozoan diseases that I've not mentioned that you may recognize, and I encourage you to go look them up. They're African sleeping sickness. Another form similar to that is Chagas disease. There's leishmaniasis, babesiosis, trichomoniasis. There's a whole bunch of other diseases that this is the reasons why we study protozoans, and because again there are some very parasitic ones that do affect humans. This will conclude part 2 of our protozoan lecture. Again, if you have any questions, please feel free to bring them to class. Thank you.